The replication of virus occurs in 6 steps. Step 1, adsorption. Step 2, penetration, also called as viropexis. Step 3, uncoating. And step 4, biosynthesis. And then assemble. Step 5, maturation. Step 6, release. So here the most important step here is biosynthesis of virus. There are 4 different types of viruses: single-stranded DNA virus, double-stranded DNA virus, and single-stranded RNA virus, and double-stranded RNA virus. Single-stranded DNA virus like parvovirus. Replication. The first single-stranded DNA virus is converted into double-stranded DNA virus by DNA polymerase, which is in the host cell. Then this double-stranded DNA, which enters into the host cell nucleus, then undergo transcription, and then which produces the early mRNA, which undergo translation to produce early proteins. This early protein is also called as progeny DNA, which has 2 functions. It shuts the biosynthesis of host cell, and it undergoes transcription to form late mRNA, and then undergoes translation to form late proteins. These late proteins are capsid and envelope, in which this early progeny DNA, which is capsulated and enveloped and then released. Now the single-stranded RNA, which is in 2 form: plus strand, which is positively called single-strand RNA, and the other one is minus strand, which is negatively called single-stranded RNA. The positive RNA, which forms early mRNA and early proteins. The single-stranded RNA, which already contains a reverse transcriptase enzyme and is converted into negative single-strand RNA, which forms late mRNA and proteins. The minus strand, which already forms mRNA and translated into proteins. Double-stranded RNA, which is converted into mRNA and proteins. This double-stranded RNA contains positive and negative single-stranded RNA, which is 1 plus strand and 1 minus strand, which acts as a template for the complementary strands of RNA, which forms the duplex. So hence 2 strands of RNA, double-stranded RNA, are formed. Viruses like HIV are replicated by reverse transcriptase method. Single-stranded RNA, which is converted into single-stranded DNA by reverse transcriptase enzyme, in which DNA-dependent RNA polymerase. Here, this single-stranded DNA, which is converted in, which is not formed directly as single-stranded DNA, but it is formed as single-stranded DNA RNA hybrid. This reverse transcriptase enzyme, which also has the ribonuclease activity, which take away this RNA hybrid. So single-stranded DNA is formed. This single-stranded DNA is converted into double-stranded DNA by DNA polymerase. This DNA polymerase also comes from this same reverse transcriptase enzyme. This double-stranded DNA also enters the nucleus and then forms early mRNA and then early protein. This early protein forms the progenome. This progenome then transcribed into mRNA and then translated into capsid and envelope, which are these proteins. This capsid and envelope are then released